Hey guys, in this video we are going to go over the beginning of the first section on biotech. Um, it's kind of a long section, so we'll just do this first one. And I have been telling you all my videos about the bird that we raised that fell out of the neighbor's nest, and mom never came back to make sure the bird was okay. So I've been raising Jack Sparrow for the last 2 weeks, and here she is. She's really Jackie Sparrow now that she's got her feathers. Um, but she wanted to join us for our meeting. So, biotechnology, the first section of biotech, is going to be using biology to advance things for us and for others, using it for technological advancement. We have been doing biotech since the dawn of agriculture, since we've been breeding plants together with traits that we liked and trying to get offspring with those desirable traits. We do that. We've been doing it with, livestock and our dog. All of that is messing with the genes and trying to engineer something that you want. But we're going to talk about biotechnology in the more modern sense when we're talking about genetics and what we can do and recombinant DNA and all that stuff. So let's get into it. Genomics is going to be all of, if we are looking at, as far as genomics, if we are looking at all of the stuff in something, their DNA that's going to lead to RNA to proteins, we are looking at genomics. So biotech, one of the first breakthroughs, I guess, as far as medicine. Um, there were many, but antibiotics is huge now, today. We didn't have antibiotics not even 100 years ago, right? Yeah. They didn't come out until the 1940s, was when we first started to at least mass manufacture antibiotics. We have been using plants and fungi for their antibiotic properties for thousands of years. But to mass produce penicillin, for example, and use that to treat diseases, that's relatively new. We discovered the shape of penicillin and what it could do back in the 40s, and then one of the very first cures was an arsenic derivative too that could treat syphilis. So penicillin is our first natural antibiotic that we were discovered, and it was Fleming that discovered it. Again, not until the 1940s. So we had all the other wars and battles and all of this other stuff without the means to give antibiotics until the 1940s. Regarding DNA, how do we play around with DNA? Well, the first thing we need to do on any cell is to extract the DNA. We need to break open its cell membrane. So how do we do this? We would use a lysing agent, some sort of buffer, some sort of detergent, soap. If we were able to do this face-to-face class, we have a lab where we extract the DNA of strawberries. And so you can just use simple things that you have at home that the detergent will break apart the uh, phospholipid bilayer on your cell membrane and bust that apart. So then you've got all the organelles inside, and you're going to have all of the things that are related to genetics. You're going to have this big mass of DNA. You're going to have, the cell produces RNA, and it's also going to produce proteins. So depending on what you want to keep that and you want to destroy the other stuff. So in this picture, we have the cell contents are treated with a protease. And if we know if it ends in A's, it's some kind of enzyme. So if it's a protease, it digests protein. So it's going to break down the proteins in the cell. And then we're going to treat it with some RNAs. So that's going to break down the RNA. So what is left over is going to be the DNA left over in the cell. So now it says it spins it in a centrifuge. We don't do that in our lab. We don't have to spin it down into a centrifuge for us to be able to isolate that DNA. We're just going to put in our ethanol and our stick in there, and you will see it rise to the surface, and then you can pull it out on your stick at this level. we've got a video that shows you how you can do this. We do this in the lab with strawberries. You can do this at home with your own cells, which is kind of cool. So if you blow up the PowerPoint video, click the link, you can watch the extract your own. And it's, again, it's simple stuff that you've got at home, hopefully, that you could do this and you could pull out your own DNA. Once you have your own DNA, we have this process, gel electrophoresis. So with gel, I'm sorry you didn't get to run one in the face-to-face lab, but you do get to play with them in the online Labster lab. So hopefully this makes sense that I'm talking at you and you've already done this lab before. But with gel electrophoresis, you are counting on the fact that DNA has a charge. And remember, DNA was really long. So in gel electrophoresis, we have to break up the DNA. You can't use, the 6 feet worth of chromosomes in a gel. You need to break it up. So you're looking at specific areas of a genome that you want to get. So you're going to use things to break up the DNA and cut it in places. What do we use to cut up DNA? We use restriction enzymes to cut up DNA. So DNA will be in fragments, and those fragments will be run through the gel. There will be big pieces. There will be little pieces of DNA, and you're going to run those pieces through the gel. So what you do to make the gel is the gel is agarose, by the way, and it's just like Jello. And so you're going to pour your gel into this handy dandy little mold like we've got right here, and it's going to sit for, I think it was like 20 minutes. While it's sitting, this little comb is in there, and it is going to create a gap. When we pull out the comb later and we remove the Jello out of its mold, it's going to have these little holes inside called wells. And so here are the wells right here, here, and here. And you are going to take different DNA samples, and you don't want to cross-contaminate. So each well will have a different little solution of DNA, and this well will have a different one, and this one will have a different one. So in our lab, we had, here's the ladder on this side, and then we had, I think it was, here was the crime scene, and then we had subject number 1, subject number 2, subject number 3, and we tried to match up the different DNA that was found at the crime scene with different people that we suspect might be the person that did the crime. Again, if they're there, though, it doesn't mean that they did it. It just means that they're there. So we have 3 different people's DNA, and we have DNA found at the crime scene that we can compare to. So every well will get a different thing. One will have the DNA ladder. One's going to have the crime scene. Another well is going to have one suspect. Another well is going to have a different suspect. So all of these different wells, you'll have all these different solutions put in there of this DNA. So now it's time to run the gel. So that mold is going to sit inside of a tray, and that's going to go in here in this machine. And you're going to pour over this TAE buffer on top of it. So it's coated in liquid. It's coated in this liquid buffer. And then you turn on the power. So when we talked about DNA and DNA structure, remember, is a nucleotide that has 3 parts. There's a nitrogenous base, C's and G's, A's and T's. There is the sugar and the phosphate backbone, the sugar deoxyribose for DNA, ribose for RNA. So nitrogenous base, deoxyribose, and a phosphate. And that phosphate has a negative charge. So DNA's overall charge is negative. So when we put a gel electrophoresis together, one side is positive and one side is negative. Where is our negative stuff going to run to? Is it going to be attracted towards another negative, or is it going to be attracted toward a positive? It's going to run towards the positive side. So our DNA is going to run, here's our negative side, here's the positive side, and it's going to run through this agarose as best it can. The little things get through faster than the big stuff. The big stuff's going to lag behind. It's not going to be able to weave its way through this Jello like the small stuff can. So you will see shorter sequences move faster than the longer sequences that get stuck behind afterwards. So the slower stuff, the bigger stuff runs slower, stays in the back, closer to the wells. The faster stuff runs furthest from the wells towards the end, and then you can compare all your different things to your ladder, to your crime scene, and see what you've got. So in this example, they've got one, it is stained with a fluorescent dye, and you put it under UV light. We use a different type of stain. The stain that's used in this fluorescent one is not great. I want to say it's a little hazardous. So what we use in our lab is a blue dye, and it looks a little bit different. But in the end, you will have a ladder and you're going to be able to compare all the different DNA strands to what you found. And did you find that same match? And so we have talked about being able to do those 23andMe kind of genetic things and upload your information into those databases. Well, I was reading this. It came out, I want to say about 2 years ago, this I'll Be Gone in the Dark book that was a true crime story about a horrible person who was attacking people in the 70s, I think possibly the 80s. And he was a murderer who progressed to, he was a rapist who progressed to murdering people. And they couldn't figure out who it was. And this was way before DNA technology. So they had the evidence, they had his DNA, but they couldn't figure out who this person was. And eventually he went to ground, and we didn't know who he was. They couldn't figure it out. And, years and years went by, and there were lots of theories about who this person was. We knew, where the attacks happened, so they could figure out, where this person probably grew up in this city, in this state, and probably moved to here at this time frame to be able to commit the crime. So they knew a lot about this person. They just couldn't figure out exactly who this person was. Well, back in the day, they were able to collect his DNA. And when this book was written, the rules were that if somebody had submitted their DNA to 23andMe or something like that, you sign this waiver. And I don't know if you've done it yet, but that waiver is 1,000,000 pages long and nobody reads the waiver. You just sign your stuff away. Well, 23andMe was trying to fight the law enforcement, saying that our clients did not allow us, did not give you access to their DNA. It's given to us, and you can't come in here and look at all this information because our clients, our 23andMe clients, did not agree to submit their DNA to law enforcement. But if the 23andMe people put their DNA out on another public site and upload it out to the public site so they can get more and more people to match up with them and find out who their relatives were, that was fair game. So in this, and I want to say the rules have changed now, and that if you're in 23andMe, law enforcement can get your information, but I'm not 100%. But back when this book was written, they were not. They were holding tight. But it turns out that the person who was doing this horrible criminal who's doing this, um, their relative put their DNA in one of those public databases to look for other people that they matched with. And so they were able to figure out exactly who this person is based on their relative's DNA. And so it turns out that they used this method to solve the crime and figure out who did this. Even though it was decades later that this person did this, they were able to figure out who the man was that was um, causing all sorts of terrible events to happen. So it's an interesting book. I think the laws have changed since then. Again, possibly that when you sign that bazillion page waiver for 23andMe, it may give them access. But it was an interesting book using real life to tie back into a cold case story. Speaking of cold case and forensics, there is also some exceptions to the rule. Because if you think that I got this DNA and it matches to your DNA, then clearly I've got the right person. But there are some instances where that's not the same. So if somebody has gone through a medical procedure where they needed a donation of somebody else's bodily product, like bone marrow, who's responsible for making all the other cells, all the other blood cells your bone marrow does. If somebody's received a bone marrow transplant, they may have cells from their transplant that are from the donor cells, and those have different DNA, right? It's not their DNA, it's the donor cells DNA. So you may have somebody else's DNA on your cells, and if say something happened and they had a crime scene's worth of DNA and they were trying to compare it to, sometimes it's not always well, this person is this DNA because that's the DNA we found here. It's not always the case. So every time there's a blanket statement, there's always an exception in there. There's an exception. So this one is talking about how this person had needed a medical procedure. They received the donation, and now their DNA is for the donor, and not always showing up as their personal DNA. So interesting stuff out there. in biotech. This is gel electrophoresis, and what gel does is it's going to separate the DNA fragments into small bits and little pits, and then you can compare those DNA fragments to what we know called the ladder and see what pieces match up. What genes do you have in that fragment, in that piece of DNA or not? If you had a piece of DNA from a crime scene and you were able to get a couple drops of blood or whatever from a scene, and that's all you had, you need to copy that as evidence. Maybe you are studying a really unique gene that this organism does, and we want to figure out what can turn that gene on and off, and we want to play with its gene expression. We want to copy it. So there is a way to copy DNA, to copy that information. So it's called the PCR. So PCR is basically your copy machine. Polymerase chain reaction is basically your copy machine for genetic information. So the way it works is we talked about DNA as a double helix. So on the first part of it, you need to denature the DNA. So you're going to put the DNA in the PCR machine and it's going to heat up. And when it heats up, we talk about how heat changes and denatures the structure of stuff. So it's going to split apart, it's going to unzip the DNA and it's going to be denatured. Then we need to, when we talk about DNA replication, we talk about how we have to have a primer. There had to be an RNA primer and the DNA pole attached onto the primer, right? And then we lay down new DNA. So that's what's going to happen here. We're going to heat up. I can't remember exactly what the numbers are, and I'm not going to stress y'all about learning what the numbers are, but we heat up really warm at step 1. Then we cool down a little bit. I want to say it's like 70 something degrees Celsius in step 1 and 50 something degrees Celsius in step 2. So we cool it down a little bit so the RNA primers can stick on to the DNA for replication. Then we're going to synthesize in step 3, DNA synthesis. So DNA synthesis is going to use a DNA pole, and that DNA pole is Taq polymerase that is coming from Thermus aquaticus, which grows in the hot springs in Yellowstone National Park, which is super cool. So normally our DNA pole is not going to be happy if we are at, I don't know, 76 degrees Celsius. It's really, really hot, and ours wouldn't work, but this one does because this organism lives in a hot springs and it's totally happy being in that really hot water. And so we use the DNA pole, the Taq polymerase, from them to now lay down the new DNA. So that's our 3 basic steps. We are going to denature, heat it up, split it apart. Then we are going to cool it down a little bit to anneal on our primers. And then the next step is going to be DNA synthesis, where DNA pole is going to attach onto the primers and lay down new DNA. So we had 1 strand of DNA here, and now we've split it apart, primers, lay down new DNA. Remember, we always lay it down 5 to 3. And so now we are going to end up with 2 different strands of DNA. And then we do the cycle again, and the cycle again, and the cycle again. All of these 3 steps are happening over and over and over again. So PCR is your copy machine for DNA. What if we have RNA? What can we do? Well, you can do something called, where did it go? Reverse transcriptase PCR. And so all of the testing that we're doing right now for COVID is every virus has a genome inside. That genome may be DNA or that genome may be RNA. With COVID, it's RNA. So how do we know if somebody has COVID? How do we know if that person is infected with this viral RNA? Well, we're going to take their sample and we're going to run it through the machine. And with reverse RT-PCR, you take the RNA and you come up with a complementary DNA strand. And using that complementary DNA strand, then we can copy that and make sure that that is the complementary DNA strand of this RNA. This person has COVID. We can figure it out from this testing. So PCR, reverse transcriptase PCR, are copy machines to make more and more copies of either DNA or using RNA and then backing it into DNA. So there are videos talking about how PCR works in more detail than what I'm telling you. I want you to know the 3 steps of PCR. It is denature, anneal the primers, DNA synthesis, laying down the new DNA. Um, know that, that DNA pole is coming from a super cool organism that lives in the hot springs, and that's why it can tolerate living in this really warm water and laying down the DNA. It is going to copy over and over again those strands, and so you end up with, instead of just this one tiny sample from the crime scene, you've got lots of it from the crime scene, and you can compare it over and over again to all the other people that are potential suspects. That is PCR. Blotting techniques. Okay, so if you were looking for a specific gene, like I want to know the gene that makes this organism glow, and I found it, and I want to see if all these other samples have that gene in it. I want to test for it. So what I would do is make a probe that matches that coding. It would be the complementary side to that coding of DNA, and it's called a probe. I can't put the probe and run it through DNA gel electrophoresis. It's not going to work that way. But I can run the gel, separate out the fragments. So that's what you do first. You're going to run the gel electrophoresis. You're going to separate out the fragments, and then you're going to blot it, just like if you blot your face and that kind of stuff. It's the same similar thing. So you're going to run the gel. You got your gel, your agarose is here, and gel is right here. You're going to blot it with this membrane. So you're going to put on this nylon membrane. We got filter paper, and then we got the membrane, and here's our membrane. So we've pulled the information out of the gel, and now it is onto our membrane here. Okay, great. So we've transferred it to the membrane. What do we do now? So once it's on the membrane, we are going to put it in a solution, and that solution is going to have the probe, and that probe is going to be flagged. It's going to be flagged with some sort of radioactive or fluorescent dye that will link in. And if that gel that you just ran has that complementary section that you're looking for, then the probe will stick. If it doesn't have it, then the probe will just wash away. So with southern blotting, you can look for specific sequences of DNA using a probe. If you want to look at RNA, you want to see what something is expressing. They're going to activate the genes, right? And they're going to transcribe the messenger RNA. What are they expressing? What genes are they expressing? Maybe I want to look at the RNA that a cell is using. Then you would use a northern blot. What if I want to see what proteins are being expressed? That is a western blot. So know the different ones. This is the technique here. Here is the technique. No, southern is going to be DNA, northern blot is looking for RNA, and a western blot is looking at proteins. So they separate out the pieces, whether it's DNA, RNA, or a protein that they have denatured. They will run it through the gel, and then they will blot it away, and then they will use that blot on this membrane and try and put the probe out for whatever information they are looking for, a specific gene, a specific segment of RNA, whatever it is that they're looking for. That's a blot technique. Cloning. We are covering so much stuff in this one section. It's a lot. So with cloning, we can do molecular cloning, and that's just like it sounds. It's the tiny molecules of DNA. So not a whole individual organism. That would be reproductive cloning is a whole organism. We have to do baby steps before we get there. So we'll talk about molecular cloning first. Molecular cloning, this is exactly what we're doing in our plasmid project, is molecular cloning. So here's our foreign DNA. In our case, we wanted to put the insulin gene in the E. coli bacteria. So the E. coli can start producing the human insulin that we need, that human insulin protein that we need, versus beforehand, we had to slaughter pigs, and we would use the pig pancreases and grind the pig pancreases up. And that's how we would get insulin. And not everybody could handle the porcine insulin, so we did this technique, and now this is how we can do it. So we take our foreign DNA, that is your DNA sequence out of the plasmid project, and you got your strip, and it's 10 pieces long, right? And then you are going to try and cut that strip as close as you can. If here's my DNA strip, I want to cut above the gene and below the gene. I don't want to cut in the middle of the gene. If I cut in the middle of the gene, gene's useless, right? We can't express a gene that's cut in half. So we want to cut above the gene and we want to cut below the gene. What cuts the DNA? Restriction enzymes, right? So we had multiple different restriction enzymes that we're looking for. We had one restriction enzyme in all of our list that will cut above the DNA strand of insulin and below the insulin strand. And then we need to be able to put that gene inside the plasmid. How do we get into the plasmid? We got to cut the plasmid. There is one restriction enzyme that cuts the plasmid in one place. If we cut the plasmid in multiple places, I can't demonstrate this because I only got the fingers. But if we cut the plasmid in multiple places, then you've just created a plasmid that's got 3 sections now instead of just one enfolded. It's got one cut here, it's got one cut here, and then you've got this middle piece. You only need to cut the plasmid one time. So you want to cut it one place and it will open up. And then you can put your gene and insert it in there. And now you've got what's called a recombinant plasmid, because those original host genes now have genes that weren't theirs. And you have recombined them into this plasmid. So our foreign DNA is going to go into this plasmid here. In this plasmid, a lot of these, we don't have it. These plasmids will have genes that are going to be indicator genes. And for this one, we have the LAC-Z gene that's indicated in yellow. And we also have antibiotic resistance genes. So we have ampicillin resistance genes, and those are in green. Those are going to be important later when we go into growing these and culturing these and making sure that what we have is what we need. So we're going to take that restriction enzyme. We cut the gene of the insulin gene before and after. We are going to take that same restriction enzyme and cut our plasmid. So we cut our plasmid, and where it cuts is right here. So it cut the LAC-Z gene. If we cut a gene in half, does it work? No. So we're going to cut the LAC-Z gene and it's going to open up. And now we have our sticky ends, and we are going to put our sticky end genes and we're going to put them in there. And now we've made recombinant plasmids. We need to know if these recombinant plasmids work. So you've put your E. coli in some nutrient media. You have put the foreign DNA floating all around in this media in the hopes that they are going to have transformation. The E. coli is going to pull this genetic information in, and it's going to be incorporated into their plasmids. But you're not 100% sure if it really happened. Did I get what I wanted in there? So let's test it and see. Let's grow this E. coli and make sure that those E. coli actually have the plasmid and they're expressing what we want. So remember, we had ampicillin resistance. So what we're going to do is we're going to grow these guys on a petri dish whose media on the inside, the agarose, is embedded with ampicillin, which is an antibiotic. So nothing should be growing on this ampicillin-coated plate unless it's got ampicillin resistance genes. And our plasmid does. Everything in green, remember, that's the ampicillin resistant gene. So that lets us know that anything that grows on our petri dish has to have that ampicillin resistant gene. So that's one way to make sure that what's growing and what we're looking at is the E. coli that we specifically want to look at. So that's one way to make sure. So now we've narrowed it down that only our E. coli are going to grow on this plate. We're not going to get staph or bacillus or anything else growing on here. It should just be our strain of E. coli that's got the ampicillin resistance genes. Now, we've got our strain of E. coli with the ampicillin resistance genes. Does it have the plasmid? That is the key feature. If it has the plasmid or not. Otherwise, we didn't get the proper recombination. It's just got the host genes and not the plasmid. How do we know if it has a plasmid? Because remember, we put the plasmid and we sliced that gene in half. We sliced the LAC-Z gene in half. The LAC-Z gene is going to allow these colonies to process the lactose, and when they do, they change color with that LAC-Z gene. It's a synthetic kind of thing that they put in there, and when they break it down, it changes color. So their colonies will grow and they will look blue. But when we put in our plasmid, we broke the LAC-Z gene. So we will grow but we're not going to change color. So everything that's growing on this plate has the ampicillin resistance genes, because nothing else, it's got ampicillin, and the antibiotics are going to kill off everything else. And then if it is white, those are the ones that have the recombinant plasmid. Those are the ones that are working and did what you want them to do. You don't want the blue colonies, because that means that they didn't take the recombinant plasmid and they didn't cut the LAC-Z in half, and the LAC-Z gene is expressing and showing up as blue. This is a lot of information to process in this one slide, but hopefully you're doing the plasmid project, and this is going to make sense to you as you do the plasmid project. You are going to take your insulin gene and you are going to put it into the DNA plasmid. You're doing restriction enzyme, there's only one, and it's going to cut the insulin above and below, and it's going to cut the plasmid circle just once, and there's only one restriction enzyme that'll do that for you. Okay, here is another animation that walks you through recombination and how it works. That was molecular. We are cloning over and over again these recombinant plasmids, and every time that these things, we're going to grow these things over and over again, and we can get more and more of the little guys that we want with our plasmids. Reproductive cloning is doing a whole entire organism. So of course, this is a little bit more complicated than what we talked about previously. With reproductive, Dolly was our first one, Dolly the sheep. And so Dolly the sheep was the first reproductive clone. She came and she was named Dolly the sheep because the nucleus, the nuclear material that we cloned, came from mammary glands. So if you were a Dolly Parton fan, this makes sense to you. If you don't know who Dolly Parton is, you're not going to get it. That's okay. Dolly Parton's mammary cells, we got Dolly the sheep. So we took the nucleus out of these stem cells in the mammary glands, and we're going to put that nucleus inside of the host egg, so our donor egg. So this donor egg, we pulled their nucleus out, their haploid nucleus out, and we put in a diploid nucleus into the egg, and then they zapped it. I'm sure it was more complicated than just an electrical zap. I'm sure they did something special. But whatever they did, this magic happened, and that stimulated the cell to now believe that it is a fertilized zygote, and then it underwent cleavage and developed, and then they implanted that blastocyst into a surrogate, and then out came Dolly. So this was 1996. When this happened, the first reproductive cloning where you were growing a whole new organism. This is the topic of sci-fi movies for a very long time, where we have been, if anybody has watched this movie, it's not the greatest movie, but if you're a Scarlett Johansson fan, it was a movie about these people, and they lived in this wonderful utopian kind of place, and they were fed the perfect diet, and they exercised, and at the end of every day they would do this raffle, or maybe it was at the end of the week, and there was this raffle, and that everyone hoped they wanted to be picked in this raffle, because if you were picked in this raffle, you got to go to the island, which was painted as this wonderful vacation place that everyone wanted to go. Yay. And it turns out that the island doesn't exist. What really happens when your name gets picked is that you go and you are slaughtered for your organs, because you are the reproductive clone of somebody else who's out in the real world. So sci-fi based on real life reproductive cloning, except we don't clone people yet. It is against the law. We'll get there and talk about all the ways that we are starting to hedge against the law. This chimeras are going to be where you are combining they're transgenics, two different things. So the whole purpose of this movie, The Island, was talking about how these people were cloned because their host, their original person, did this as insurance in case something happened and they needed an organ transplant later. And if they did, then they had their clone sitting in this area, and that they would harvest the organs from the clone, and it's a self clone. You don't have to worry about it. What if we could grow our individual organs that we need in another animal? Could we do that? So that started this one here with the human pig chimera. They started it as rats and mice to see if, I can't remember if it was a rat spleen and a mouse or a mouse spleen and a rat, but they were able to grow the organ from one in the other animal. The other animal could grow that and develop it. So then they started to see, well, if we could do that, what if we could put human stuff in a pig and make a chimera that way? And would that grow and develop? And they did. They were able to grow, and they stopped it. I can't remember how many cells it was. If it was like a morula stage or a blastula, I can't remember if it was like 16 or 32 or 100, but they stopped it before it got too far in development. But they did this human pig chimera where they were trying to use it for growing people's organs that they need. So we are pushing the envelope all the time. In the last couple of years, we're going to get to CRISPR and talk about that too. So genetically modifying, I was telling y'all, we've been doing this for millennia with agriculture, with livestock, with the dog that I have in my bedroom right now. He's running around like a crazy dog, growling at invisible gremlins. Those, we modified them for the traits that we want. When we're specifically talking about modifying their DNA as a recombinant, that's a genetically modified organism. So that's GMO. If we are using genetic information from an organism that's not the same species, they are transgenic. So we have put human DNA in a pig, rat DNA in the mouse. Those are transgenic. Other species put into the host species. Other terms on here: reverse genetics. So in the past, all we have were phenotypes. And so we were trying to figure out what genes gave us those phenotypes. But now that we can find the DNA and mess with the genes ourselves first, now we can go from genotype to phenotype and figure out, what does that gene do? If I tweak this gene, if I mute this gene and I block it, what happens to the phenotype if I do that? So now we are doing reverse genetics. What if we do gene targeting? Like we said, we're going after the specific DNA and we want to mute it. We want to stop its expression. We're going to play around with its expression, either deleting it, putting in some sort of mutation. This is gene targeting. So sometimes we have a gene that's not right. It's got a mutation. Something is happening and it is bad. There are a number of genetic diseases that if we could possibly fix the mutation going on with them, then that person would have a normal, healthy life without that mutation. So what if we could put that gene and fix it? What if we could cut it out and fix it? So there's gene therapy. So if you had a genetic test and you knew that you have this going on with you, the pros sound amazing. If I could get rid of this genetic condition and no longer have to suffer with this issue for the rest of my life, sign me up. But with genetic testing, with gene therapy, the way it works is they are going to take the good gene. So your gene is misfiring. It's got a mutation and it's, coding for the wrong protein. Maybe you got CF, and CF codes for the wrong protein, and that leads up with the mucus buildup on all of the mucous membrane surfaces in your body. If you could go in there and cut out that gene and change the mutation and put in the good gene, what you do with gene therapy is they're going to put it inside of an adenovirus, this guy right here, and they're going to modify the genome inside that virus to include your good human gene. And then you're going to infect the cells with that virus, with the adenovirus, in the hopes that the adenovirus is going to infect that cell and do what viruses do. They hijack the cell, right? They put their genome in our cells. So that's what you're hoping with gene therapy, that you're going to put a good gene inside of a virus. That virus is the vector that will deliver the good gene to your genome. And now you will be able to have the healthy, normal life because this gene will be put in there in the place of or override the bad one. There are some cons in here. Initially, we thought that this was, great and there wouldn't be any issues. And so they performed this on someone, and I can't remember his name. We talk about it in micro class, but this person had signed up for it, and 4 days later, he was dead. And so he suffered from consequences and side effects of this. So here are the different side effects. That adenovirus, the virus that is the vector that brings you the good genes, can cause an inflammatory response in the immune system that leads to shock, that leads to organ failure. It's a cytokine storm is basically what happens. Or that vector infects cells that are not targeted. If you're at CF, your mucous membranes, those are the cells that you want to target. And if you are able to infect the cells in your respiratory system, you're not infecting the cells in, your GI tract or your reproductive tract or otherwise. You're only infecting the ones that you want to infect. That's the plan. But what if the viruses start infecting cells that weren't targeted, and it starts just going crazy, infecting all the other cells? What if the virus that you have modified so it does not hijack your cells and replicate and make more of its own virus? What if it reverts back to being infectious and it causes the patient to be infected with the adenovirus and they get sick? That's not good. We don't want to make somebody's issues worse. Or that gene that you put in to override the sick gene and the good gene somehow unintentionally stops a different gene and inactivates another gene. So gene therapy sounds wonderful, and it has the potential to be life-changing. But there are some side effects associated with gene therapy. It also leads to ethical concerns if we are going to modify genetic information. Who decides what needs to be corrected and what doesn't need to be corrected? Should we use it for everything? What if I just want to change cosmetic reasons? I want to change hair color or eye color or make myself more muscular, whatever that cosmetic change is that worthy of gene therapy? What if we're messing around with fetuses and we make designer babies? Does everybody get gene therapy, or just the rich? How's it going to work? And then who's responsible for regulating and policing inappropriate use of it? Somebody breaks the rules. Somebody made a designer baby. Who holds them accountable? Who's in charge? So CRISPR is a relatively newish thing on the scene. In the last decade-ish, we have been playing around with this. And I have this video here. It's Bozeman Science Guy. He's got some great videos, and he talks more about the origins of CRISPR, how it works, but it's pretty much gene editing, where you use the CRISPR-Cas9. It will cut out the undesirable gene and put in something else for you. And so it is gene editing. You can watch the video and learn more about CRISPR. It's not even in the textbooks. I don't know why, but it should be. So you can watch the evolution of CRISPR in the last couple years. We have articles talking about how we could use it to fight cancer. If we know what's getting expressed, what cancer is causing to be expressed, maybe we can mute that. If we knew that this embryo has this lethal genetic condition and it's not going to make it, what if we went in there and we altered it and then we took out that lethal gene? Could we do that? Is that ethically okay to do? There is a person out there that did that, not exactly, but he modified some embryos, and there is a receptor that if you don't have this receptor, you cannot bring in, it's harder anyway, to bring in HIV into your cells. So if you lack this receptor, HIV literally it can't get in. This person modified those embryos to specifically make it so they don't have that receptor on those cells. They don't have the receptors. And then I'm not sure exactly what happened. It was really shady. If he talked to the parents, if he didn't tell them what was going on, but those embryos were implanted, and the mother gave birth to these twins that have modified at the stem cell, at the embryo level, modifications. That was not, it was beyond what we are supposed to be doing at this time. We are not doing stem cell at the germ layer level of modifications. Previously with our gene therapy, these were already adults with their somatic cells already formed. Genes at the stem cell level, that means that it's being transferred to all the other cells in the rest of the body, and it will be transferred to these girls' offspring too. So this is a huge, huge deal. This was, I want to say this was 2018. So it's a couple years old, but CRISPR is the new thing to look into. It's exciting and scary all at the same time. Vaccines. I can't say enough to stress about how important vaccinations are. They are going to help all of us. Hopefully most of us have vaccines, but what a vaccine is and how it works is that they are going to modify. They're going to take a piece of, and that could be a piece of the bacteria, like the outside or the antigen on the bacteria. It could be a modified piece of the virus, but you're going to take that, it's an antigen. You're going to take that and you're going to expose the person to that piece, that modified, that dead virus that you have changed. And we expose you to that substance, that piece, that antigen. And then your immune system will now, your immune system is going to be ready. And if it ever sees that piece again, yes, I see, yes. If it ever sees that piece of whatever it is again, then it will know it and it's waiting for it. And if it sees it again, it will attack. So that's what our vaccinations do. They are ready. If you ever get exposed to mumps, measles, rubella, polio, your body is already ready and waiting, and it will take out those diseases so you don't get sick. That's how vaccines work. It is not an entire whole organism that is infecting you with it. That's not how it works. It's modified and tweaked so that it's not going to come back alive inside of you. You're not shedding off this organism if you get a vaccine. So there is a video here that talks about how they work. I know there is a huge anti-vax movement out there, so I want y'all to understand how it works, so you can educate others about how vaccines work, because not everyone can get vaccines. I mentioned my brother-in-law went through this terrible chemo that basically took out all of his stem cells. It did. They took out all his stem cells, they harvested his stem cells, they irradiated him with this chemotherapy, and he had to start from fresh. He had no immune system after that point. So he had to go back and get, once his white blood cells came back and they bounced back, he had to go get revaccinated because he didn't have any immunity anymore. There are people in the population that they cannot get vaccinated, and they're hoping on other people's herd immunity that they will be okay. So I do want y'all to understand what vaccines are, how they work, how they provide health. I know in the era of COVID, we are all hoping that there is a COVID vaccine, and I know people are working on it. It's just not out yet. It will still take a long time to trial it. People are in test trials now for it, but it takes a while to make sure that what you give people is going to be okay and safe to use at the same time. So vaccines are our friends. Antibiotics are our friends. Does an antibiotic work on COVID? Antibiotics are not going to attack viruses. They attack bacteria. So if you have the common cold, you're not going to need an antibiotic. That's not going to treat the cold virus. It's not going to help you. But if you have an infection of strep, if you've got strep throat, an antibiotic is definitely going to help you. So all of these are biotech. And we talked about making insulin as a hormone. All of these are great advances that we have in the form of biotech. Transgenic animals. We talked about making the transgenic bacteria where we put in the insulin gene in there, and we changed it and made it so that E. coli is now producing human insulin. That would be a transgenic prokaryote, What if we made transgenic eukaryotes? We do. We have made goats that are modified to where when they are producing their goat milk, they are also producing anticoagulant factors that people need. We've modified these mice to where if they have this recombination, there's also, remember how we talked about that LAC-Z gene? It would or wouldn't work, and that was our indicator to know that the plasmid went in or it didn't, it was successful or it wasn't. The same with these mice. If this mouse glows, then we know that whatever else we put in that is working, that modification is working, because mice don't normally glow when they're exposed to UV light. So we have made transgenic animals. We have made transgenic plants. How do we make a transgenic plant? Well, it's hard to get the information into the plant. The plant has a cell wall and a cell membrane. So what we can do is use a bacteria plasmid. So there's a bacteria. There's a bacteria that infects plants. It's called Agrobacter tumefaciens. And what it does is when it infects plants, it causes the plants to grow tumors. And so the agriculture does not like this bacteria because it depletes their crops. The plants are still alive, but they're making tumors and they're not producing what they should be producing instead. So with this Agrobacter tumefaciens, here's their plasmid and here's the tumor DNA region. We're going to cut the tumor DNA region and we're going to stick in a gene of interest, whatever gene it is that we're looking for that we want to have in the plant. We'll put that gene in here, and now we've stuck it in between the tumor gene. So we've broken the tumor gene. We're going to put this plasmid back into our tumefaciens bacteria and have that bacteria infect the plant. So now it's gotten into the plant cell. The plant cell incorporates that plasmid, and now the plant cell is doing what it was infected with. If it was infected with a plasmid that did not have the new gene in it, it'd make tumors like normal. But since we put in this new gene and cut the tumor gene in half, now it's going to do whatever that new gene wants it to do. So we have BT corn, and that's coming from a chemical that's produced, a toxin that's produced by Bacillus thuringiensis, and that makes that bacteria produces a toxin, and when insects ingest the toxin, it causes crystals in their GI tract and they die. So what if we could put that toxin into the plants, and now everything that eats that plant, as far as an insect, will get crystals in their GI tract and die. So that's what they did. They made BT corn. I think it's Monsanto possibly that has the patent on BT corn. So we have made transgenic plants. We put this crystallizing gene from Bacillus thuringiensis and we put it inside this plasmid. It gets uptake by the plant, and we have genetically modified. We made a transgenic plant using bacterial genomes inside of the plant. We've done this before with the flavor saver tomato. I think this was in '94. And so those genes were to slow its decay and to change the flavor. So it was supposed to be tastier and it was supposed to last longer. So you could get it, I don't know, across the United States and it would still look nice and ripe and yummy, delicious. There were some issues with it on its rollout. I don't think it was able to be successful, the flavor saver tomato. So that's not out there. I want to say BT corn is still out there right now. So these are transgenic plants. This has been a huge, long video. I'm going to stop it right here and we'll do another video on the other sections.